Today, seminar is hosted by the biology department, as well as the IMSD program at UMass Boston. And in addition to having our honored guest and a special guest, we're also going to have a special introduction. Monica Robertson is an IMSD scholar on campus. She's a biology student, and she will do the honor of introducing our guest. Before you do, Monica, I want to remind everybody that at the end of the seminar, you could put your questions, or maybe during the seminar, you could put your questions in the Q&A, and concluding the presentation, Dr. Murray will answer questions then. Monica, take it away.
Okay, well, it is truly a pleasure to be able to introduce my research mentor, Dr. Andrew Murray. He's a Herschel Smith Professor of Molecular Genetics, a Professor of Molecular and Cellular Biology, and Director of John Harvard's Distinguished Science Fellows Program at Harvard University. He received his BA from Cambridge and his PhD from Harvard, where he worked with Jack Szostak to construct the artificial chromosome. In his postdoc work, he worked with Mark Kushner at UCSF to show that cyclin synthesis and destruction regulates cell division cycles. He also was a faculty member at UCSF, where before he joined Harvard's Molecular and Cellular Biology department. He also has published hundreds of articles and received numerous awards, including the Professor of Howard Hughes Medical Institute and the member of the American Academy of Arts and Sciences and the National Academy of Sciences. Even with all of that, he has taken special care in developing and teaching mentorship, including many former trainees that have become faculty. This also includes an incoming faculty member here in our biology department, Dr. Juliette Girard, which we're all very excited for. With that, Dr. Murray, we are very excited for you to join UMass Boston's community today, and we look forward to your talk.
Thank you so much, Adan and Monica. It's really an enormous pleasure to be here. I'm sorry I can't see all your faces, and I'm sorry you're going to have to wait for Q&A to the end, because normally I encourage people to just stop me when I say things that don't make sense, but you can tell me all the things that didn't make sense afterwards. So I'm going to talk to you today about a trip to the evolutionary repair shop. And so I'm going to start by just reminding you that here's a picture of part of the sky. And there are a lot of stars there, and there's some planets. As long as none of those planets have living things on them, there's no non-theological purpose to what you're looking at. Okay, and what I mean by that is everything there is just obeying the laws of physics. And so the word purpose doesn't really make sense, because purpose implies something with a desire to do something. And organisms, however, if there's a planet with life like there is on Earth, organisms do have a purpose in an evolutionary sense. They're vehicles to disperse genes through time and space. So this is a famous but fictional vehicle for doing that. This is the USS Enterprise. Okay? And I'm going to remind you that evolution really does happen. So this is a metaphor. This is an example of what a typical human cell looks like. It behaves a little bit like a very prim and proper Victorian woman. It's born and lives and dies all very much close to the same place. It follows an extremely rigid set of social rules. And these cells would often die rather than misbehaving. Okay? A series of mutations, at least 5, can turn it into a cancer cell. Okay, with an appropriate cartoon metaphor here. And these cells are very careless with their chromosomes. They're genetically unstable. And this is an example of evolution in a human being's lifetime. Now, the cancer cells, in a sense, are rebels without a cause, because they can't be passed on indefinitely into future generations, because they will eventually kill the body that they inherit. And when I do this in person, I always say if there's someone at the end who can identify both of these people, these are actual individual humans who've lived, I always bring a 20 and I'm prepared to give it to a person who can say who each of these people are. But you have to name both of them. So if you want to try in the Q&A saying who you think both these people are, you can give it a whirl. Okay. So evolution depends on inheritance, that offspring look like parents because they inherit some of the same genes. It depends on mutation, so we constantly generate genetic variation, and it depends on selection, so some genotypes leave more progeny than others. We have trouble understanding evolution because it's dominated by a succession of very rare events, okay? And it's the relative probability of different classes of those very rare events that determine what will happen. And unless we play the lottery, which I actually do occasionally, we're not very good at dealing with very rare events. And in fact, I'm not that good at dealing with the common one, which every Tuesday and Friday I wake up and like, damn, didn't win again. And the other reason that we have trouble is that our ability to infer what happened in the past, either from fossils or from the evidence of DNA sequences, is imperfect, okay? So we're missing one simple thing. So if you're a Doctor Who fan, this is the TARDIS. This is the vehicle that Doctor Who, the British TV science fiction series, uses to travel through time. So if we had this, we don't need me because we can just go back and see what happened, but we can't, okay? So our substitute for Doctor Who is what I call experiment-based speculation. So we do laboratory and evolution in the laboratory and try and figure out what happened in retrospect. And so what I'm going to talk to you about today and what I mean by the evolutionary repair problem is, here is a famous car. This is the Citroën DS. It's on everyone's list of the 10 most cool and groovy cars of the 20th century. And one of the reasons it's there is it had self-leveling hydropneumatic suspension, so you can drive it like this with missing wheel, okay? And if cars evolved and those three-wheel Citroëns gave rise to progeny and there was adaptation, it would probably look like this. And so we're interested in the biological equivalent of this, okay? And so you could ask, well, why are you doing evolutionary repair? You know, isn't life perfect and organized and not needing repair? And the answer is that during the history of life on Earth, all sorts of important things have disappeared, okay? So this is a picture of a single-cell protozoan called Giardia lamblia. If you drink water that hasn't, if you go hiking in the West in particular, and beavers have been pooping in the water, it's a very unpleasant, long-lasting intestinal parasite. And there's this thing that eukaryotic cells use to escape from mitosis. So they have to destroy particular proteins to escape from mitosis. And that machinery is called the anaphase promoting complex. And it got discovered in frogs and it got discovered in yeast. And that was good enough for most people. It was like, oh, this is a universal eukaryotic protein complex. And then it was discovered in a whole bunch of other things. And eventually some people got around to sequencing the DNA of Giardia. And they tried to publish a paper which says Giardia is sort of weird because it's lost a whole bunch of things, including the anaphase promoting complex. And when they sent their paper off, the reviewers' response was, you people are not good at sequencing DNA. Of course it has the anaphase promoting complex. Go back and do the job properly. But they eventually convinced people that it's true, it doesn't. So here's something that until we looked at this organism, we thought was universal and essential for getting cells out of mitosis. And these guys must have figured out some other way of solving the same problem. And things get rearranged. So I'm going to talk about the machinery for replicating DNA. So all cells that we know of have a genome made of DNA. There was a last common ancestor that replicated the DNA. And so you might think that the details look surprisingly similar. And so we're going to study this problem in the budding yeast. And we do that because they divide every 90 minutes. They can proliferate both sexually and asexually. And that asexual sexual switch is actually useful to us especially in evolution experiments, for tracking down the mutations that cause the phenotypes at the end of the experiment. It has excellent genetics, both as haploids and diploids, and mate them with each other. And it has spectacular genome-based tools for mutations. So amongst all eukaryotes for sure, and possibly even more so than E. coli, the community of people who worked on yeast have developed a spectacular set of tools to analyze and find and create mutations. So when people who are as old as me talk, it always means that someone else did the work. Okay, and so this is the someone else. This is Marco Fumasani, and actually, what's sort of spectacular is he was a postdoc who worked in the lab, and next week he's flying to Lisbon to start a job there. And this evening, after your guys' happy hour, I'm hosting a virtual happy hour to say goodbye to Marco. And so he did his PhD in Italy working on DNA replication, but sort of working on the molecular details. And so here's a picture of DNA replication, and there's double-stranded DNA here. And there's one strand where the new strand is being made continuously because it's going in this direction. The other strand, it has to go the other way, so it has to keep getting made in little chunks. This blue thing is a helicase, which unwinds the two strands of the DNA so you can do the replication. The orange guys are the polymerase. This red thing is a clamp that keeps the polymerase from falling off the DNA. The yellow octopus, if you like, is a machine that loads the clamps on and helps to hold things together. So this is what it looks like in E. coli, where people really worked out all the details. And here's what it looks like in you or in yeast. And if you look at this diagram casually, you're like, oh yeah, it looks just the same. So I'm going to draw your attention to some differences. Okay. So here in E. coli, the helicase is going along the strand that you have to make in chunks. Here in us, it's going along the other strand. In E. coli, the helicase is 6 identical proteins all in a ring. In us and in yeast, it's 6 different proteins. They're all related to each other in a ring. Okay? And in E. coli, the primase, the thing that makes little pieces of RNA, which is how you start before you make DNA, is hooked to the helicase. And here it's hooked to something else. So even though the sort of fundamental logic of DNA replication has been conserved, the details have changed. And yet somehow those details had to change while organisms were successfully replicating their DNA. Okay? It's sort of modifying your spacecraft as you fly it. Okay? And this thing here, the green octopus, is the one part of this machinery where by looking at the sequence of the protein, you can unquestionably argue that the green octopus has a common origin in eukaryotes and bacteria. The other parts, it's less clear. Okay? And I just explained the difference between the helicases and the two organisms, that there are important differences in the actual composition of this ring, and the ring is moving along a different strand. And so what Marco wanted to do was to not break utterly, but to damage the DNA replication machinery. So what he did is do the geneticist equivalent of putting a bullet through a particular part of it. And what that means for a geneticist, excuse me, is you just delete the gene. Okay? So this is a slightly more detailed view. Here's that helicase, that 6-membered ring. It has some friends. Here's the thing that's making those little RNA primers. And here is the protein that Marco eliminated. It's called CTF4. Okay? And previous people had shown that when you remove that protein, the cells grow poorly. They have trouble holding their sister chromosomes together all the way until the time when the chromosomes are supposed to separate at the end of mitosis. They have defects in recombination, and they're less able to withstand DNA damage. Okay, and so here's Marco's approach. He removes this gene, and he takes a population of cells that lack the gene. He allows them to grow for 24 hours, which is about 10 cell divisions in yeast. The next morning he comes in, he dilutes them 1 to 1,000 into fresh medium, and he just does this every day until they've gone through about 1,000 generations. He measures their reproductive fitness, okay? And reproductive fitness just means how fast they grow relative to wild type. So and this number here is the percentage difference in the growth rate. So wild type, the growth rate of wild type is obviously identical to itself, so there's no difference. The strains where he had removed CTF4 grow about 28% slower. So that's a fitness defect. And when he evolves them, they increase. And instead of growing 28% slower, they're now growing sort of close to 10% slower. And last but not least, we take those populations and we sequence their genome. So we sequence end to end the whole yeast DNA sequence. And we look for cases like this, where we see the same gene mutated in many populations, even more so with this green gene. And if the gene's been mutated that many times, we can calculate that it's unbelievably unlikely that it got mutated by chance. And that suggests to us that mutations in that gene actually help the cells get fitter. And therefore, understanding what that gene is going to tell us something about how the evolution worked. Okay? But that sequencing implies, but it doesn't demonstrate that mutations improve the fitness. And so we then do experiments to test. So we, at the end, we think we know which mutations make the cells fitter. And then we engineer the strain where we remove CTF4. We engineer those mutations in and ask, do they really increase the fitness? Okay. So Marco did all that DNA sequencing, and he found a whole bunch of genes that were mutated. So each circle here is a gene. And the diameter of the circle tells you how many times in parallel populations it got mutated. And all these little gray links between the genes are things that other people doing various other sorts of experiments have suggested are genetic interactions between genes. Okay? And so an obvious question is, what the hell are these genes doing? So the blue guys are all genes involved in DNA replication. The green guys are all genes involved in holding sister chromosomes together or breaking the linkage when it's time to segregate them. The orange guys are part of this thing called the DNA damage checkpoint. And its job is, if a cell has received DNA damage, it is to stop the cells before they segregate their chromosomes so they have a chance to repair the damage. And the last set of guys here is the only group I'm not going to talk about today. These are things involved in how you wrap the DNA around histones to make chromatin and how you dynamically change the chromatin. Okay. So now if we look in detail at which genes are mutated, this is a cartoon of the replication origin. So this is the place in eukaryotes where replication starts. The DNA is these black strands. This little bubble is because replication is about to start. There's a whole bunch of different proteins. They just got themselves given different colors. But just one thing for reference. Here's that helicase again, the 6-membered ring. They're going to go this way and that way. And here are all the genes in that machinery that got mutated. The number in parentheses is the number of independent experiments they got mutated in. And if it's in red, it means it's a gene that's essential, a gene you can't do without. And that's the replication origin. Here's a picture of the replication fork. Here's our friend the helicase again. And again, there's a whole bunch of proteins that got mutated. Some of them, this guy got mutated 6 times. And again, the red ones are essential. Okay. And so we're going to zoom in a little bit on the replicative helicase. So this is the helicase. The gray hexamer is the helicase itself. The other parts are its friends. All these genes are essential, and lots of them got mutated multiple times. And I'm going to talk a little bit more about mutations in these 2 genes that are called CDC45 and SLD5. Okay, so here's a picture of that helicase. So this is the work of other people. This is X-ray crystallography. The DNA, you can see the 2 strands wrapping around each other, and it's sort of going into the screen. Okay. And then here's the helicase wrapping all the way around it. And this is one friend, CDC45. And this is a complex of 4 other friends, including that guy SLD5 I mentioned to you. This is the same picture, and this just shows where all the mutations occurred that we saw in our experiment. And this guy down at the bottom here, that's just one mutation. It's a mutation that cut off the C terminus of one of the proteins. Okay. And that little arrow goes to where CTF4, the protein that we removed, would normally bind. So one of the things we can do is we can say, well, okay, so we found these mutations. What do they do? So this is our ancestor, which is missing CTF4. This is an evolved population, and it has a mutation that's been selected. So what Marco can do is just take that one mutation and engineer it back into the ancestral strain that lacks CTF4. Okay, so that's just a little piece of genetic engineering. And now we can ask what that does to the fitness of the cells. So and sorry, there should be words up here. This is the fitness increase going up in percentage relative to the ancestor, which is missing CTF4. So we test it against itself and make sure that we get a value of 0. We put in this mutation that occurred in the evolved strain. So this is a mutation in that protein SLD5. And what we find is it increases the fitness by about 9%. And here's one in CDC45, and it increases the fitness by about 5%. So these mutations really made things better. And some of them have a really intriguing property. So this is the same data as on the last slide. Now we actually have words on the y-axis. So this is a strain lacking CTF4. This is when we put this mutation in, it got 9% fitter. But we can do a sort of complementary experiment. We can take the wild type strain that we started with that does have CTF4, and we can put the same mutation in. And now, instead of the fitness going up, it went down. So the mutation helped our crippled guy do better, but it made the wild type do worse. And the suggestion is that we somehow turned something down, and that helped the wild type cell. Okay? And so we can ask, what are these mutations actually doing to DNA replication? Okay? And we can do that by measuring when different parts of the genome get replicated during S phase, which is when the cells are replicating DNA. So we have mutations. This guy here, this is SLD5, and so that's where the mutation is. This is a different mutation that inactivates a gene called IXR1. And IXR1's job is to help the cell make the nucleotides that are required for DNA synthesis. And so the suggestion is that maybe both of these things are slowing down the rate of DNA replication, and in these compromised cells, that helps things. okay? And so we can actually do an experiment where we look at when in the cell division cycle the DNA gets replicated. So this is looking along a part of chromosome 13, and we have ways of measuring when the DNA gets replicated. So earlier is better, so the time axis is sort of backwards, if you like. So this is 15, 20, 25 minutes. And so replication starts here and it moves off in either direction, which means these parts get replicated later. This red guy here is the guy that's missing CTF4. So you can see two things. First of all, replication starts much later. And secondly, there are parts of the chromosome which haven't even gotten replicated by the end of this experiment. So replication is seriously screwed up. When we add to that ancestor the mutation in SLD5, what happens is that now replication starts earlier and you don't have these horrible parts that you haven't completely replicated. It's still slower than the original wild type strain, but we've definitely done something to make things a little better. Okay? And this just highlights this part where we're clearly having problems, because in our experiment, we haven't even noticed that it got replicated yet. Okay? So that's DNA replication. So the next thing we notice when we sequence, or that Marco noticed, you can sequence along a chromosome and you sequence little 100 kb chunks and you sequence them again and again so you make sure you don't screw up. And you can count how many times you sequence the same part of the genome, and you come to these weird places where you have extra copies of part of the chromosome. Okay? And in 14 different populations, this part of the chromosome here, of chromosome 4, got amplified. Okay? And in 7 populations, a part of chromosome 5 got amplified. So now you get to play a guessing game. You ask which genes are on those 2 parts of those 2 chromosomes, and is there anyone there who looks like they're a likely suspect, okay, based on what geneticists already know about the genes. And fortunately for us, the guessing game went fairly well, because on chromosome 4, there's a gene called SCC2. On chromosome 5, there's a gene called SCC4. They make 2 proteins that bind together to form a complex. And that complex has a job. It's to load this thing, which is a ring, a protein-based ring, onto the chromosomes called cohesin. And you load that ring on so that when the 2 chromosomes have been replicated, so you have 2 copies, the ring encompasses both. So now the sister chromosomes are held together and they can't fall apart. Okay? And so what we already knew about mutations in CTF4 is they don't load cohesin as well as normal. And so Marco's hypothesis is we amplified these 2 genes so we could make more of the machine that loads the cohesin ring onto the chromosome. We load more rings, and if you load enough, it doesn't matter if some of them fall off. Okay? And again, you can ask the question of, did it really help? So here's our relative fitness again. Here's our ancestor who lacks CTF4. Here we put in some plasmid that doesn't have a useful gene on. Here we've put in an extra copy of SCC2. Here we put in an extra copy of SCC4, and here we put in an extra copy of both of them. And what you can see is when you put in an extra copy of SCC2 and when you put in an extra copy of both, the cells get fitter. And you can ask, is that because the chromosomes are being held together better? Okay, so here, what we're scoring is the fraction of the time that the 2 sisters fall apart early, which we can see under a microscope. So here's wild type. Here's the cell that's missing CTF4. And as we put more copies of the cohesin genes back, so this is 2 copies of SCC4, 2 copies of SCC2, 2 copies of each. Each time we put more stuff back, we reduce the fraction of the cells that separate their chromosomes too early. Okay? So last but not least, we get mutations that break the DNA damage checkpoint. Okay? So the normal job, as I said at the beginning, is if you've got damage to the DNA that occurred in S phase or G2, what you do is prevent the cells from separating their chromosomes until that damage is gone. Okay? And if you knock out the checkpoint, that stop sign is replaced by a green light and the cells charge through. And what Marco found multiple times is he had mutations that knocked out this protein called RAD9, which is a critical part of this checkpoint. And there's an interesting question, which you're welcome to ask, and you will engender some speculation about how can it possibly be true in a cell that's having trouble replicating its DNA that removing a gene that is supposed to make cells slow down and wait if they have problems replicating their DNA is actually making them fitter instead of killing them. Okay? So the last question is like, okay, you guys did good. You've tracked down all these mutations. Is this all that's really required to produce the fitness increase that occurred in your whole big evolution experiment? So let me explain the vertical scale. So now we're measuring the relative fitness compared to a cell that's lacking CTF4. Okay, so that's where we started. This bar here is the fitness that the different evolved populations got to. And this bar here is where wild type strains that have CTF4 would be. Okay. So each of the vertical lines is a mutation where we put into the CTF4 delete one of the things that we found in the evolved strains. So this is extra copies of SC2. This is a mutation in SLD5. This is killing RAD9. This is killing IXR1. All those guys individually make cells fitter. Okay, so those were the singles. So now we can do the doubles. And once we do the doubles, we have a color code because we can make a prediction for how fit a double mutant should be. So if one of the mutants makes it 5% fitter and another mutant makes it 5% fitter, the naive prediction is if you make the double mutant, it will be 10% fitter. And if that's what happened, we'll color it white. If it makes it 15% fitter, that is the double mutant is better than you would expect, we'll color it green. And if it's less fit than you expect, we'll color it red. Okay? So these two guys here, RAD9 deleted along with SLD5, and come back cursor, IXR1 deleted with SLD5, and also RAD9 deleted with IXR1. All those guys are less fit than you would expect from the contributions of their individual mutations. Then we can make the triple mutants, okay? And these two, and especially this one, are more fit than you would expect, so they're green. And these two, both of which have both SLD5 and IXR1, are substantially less fit than you would expect. And here's the quadruple, which is all of them, and it's less fit than you would expect. And these red arrows along the bottom are all the guys who have mutations. So it's this one and these three down here, are all the guys who have both mutations in IXR1 and SLD5. So those guys together are bad. And what we think is true is that both of them slow down the replication fork, and that slowing down the replication fork helps. But if you slow it down too much, it's bad. And the interesting thing is that when we look in individual populations, and especially clones in those populations, we never saw mutations in the same cell in both IXR1 and SLD5. So the short answer here is it looks like we can recapitulate 1,000 generations of evolution with a small number of mutations. And that for the most part, emphasis on the most part, the fitness you get from multiple mutations is roughly the sum of the effects of the individual mutations. Okay? So what did we learn from all this? So the first thing we learned is that conserved molecular pathways are plastic, that we can fiddle with the DNA damage checkpoint, we can fiddle with sister chromatid cohesion, we can fiddle with DNA replication, and we can not only fiddle and get away with it, but we can actually make cells do better, at least if they were these genetically compromised cells to start with. So the take home is that cells recover after damaging DNA replication. They do that by modifying conserved molecular processes. And that might well have consequences for genome evolution, because if you start to change the way your genome behaves, and now you start mating to people who don't have those same changes, you may have trouble reproducing properly, and you may have started to set up the genetic barriers that separate one species from another. Okay. And so here's a sort of overall summary. Cells adapt to perturb DNA replication. The replisome, that's the whole complex of proteins that is responsible for the replication fork and the progress of DNA replication, can accept mutations in essential genes. And the other conserved pathways have been turned up and down. And so the very last thing to say is, are we learning anything about evolution that happens outside the lab? And so there's a different sort of experiment that we've done that I haven't talked about today. And that's an experiment to try and persuade yeast cells to do novel things. So we've asked yeast cells to become multicellular. We've asked them to develop a circadian clock. Those experiments work. We can track down mutations. We can find what the mutations are, but by and large, we don't understand them. And different parallel experiments don't look very much like each other. Whereas when we do these evolutionary repair experiments, by and large, the sort of evolutionary trajectories, different versions of the same experiment take are similar. It looks to us from doing a bunch of experiments like this now that repair requires mutations in several parallel pathways that come together to control an overall cellular process. And as I said on the slide where we mixed all the mutations up, the interactions between the mutations are largely, but not exclusively, additive. And the most interesting thing from all of our experiments is that most of the mutations we get are mutations that make proteins either not work at all or work worse. And it leads to the idea that when you're under really severe selective pressure, for sure in the lab and possibly in nature, you get mutations that damage genes, okay? And it leads us to this question of, is it true in nature that novelty is produced by gene inactivation? And so your first response should be to be wildly skeptical about this, okay? And I would encourage that skepticism. But there's a sort of interesting experiment that humans have done, which is an accidental evolution experiment, and that's the domestication of crops. So for roughly 10,000 years, human beings have been domesticating crops, so teosinte, which became maize in Central America, the domestication of millet and other grains in Europe. And so now scientists, plant biologists, are able to go back to those ancestral undomesticated species and the stuff that makes the food that ends up on our grocery shelves today, and ask what are the mutations that changed the wild thing to the domestic one. And there's a paper where they looked at 60 identified genes which have mutations. For a third of those genes, they're loss of function mutations. And that means they're transposons that jumped into genes got deleted. So in the original people who domesticated crops didn't know about Mendel, because Mendel was 10,000 years in the future. And, you know, it wasn't a laboratory. There were wild animals running around their fields. So this is sort of a more or less in nature evolution experiment to produce novel traits and increase crop yields. And interestingly, at least a third of the mutations inactivated genes. And those are only the mutations where you can tell by looking that it inactivated a gene. If you just substitute an amino acid, until you do experiments, you can't tell whether that inactivated the gene or not. So one of the ideas I'm really interested in exploring in the future is how much of evolutionary novelty has been produced in circumstances where you need to change things really fast under high pressure, and discover it's easier to do that by breaking things at the level of genes and by making them. And with that, I will stop here and be very happy to answer questions.
Thank you very much, Andrew. This is amazing, incredibly interesting. Maybe what I could do is I could start with the first question, and then others in the audience, you're welcome to put your question in the Q&A. So I'm very intrigued by some of the things that you said at the end, which maybe you've have already done or are you going to do, but I'd like to hear some more thoughts about this. So in running these evolution experiments where you have, in some ways, a controlled condition, is probably normal media, it's not stressful conditions for these cells to evolve. Would we have the same players lost in evolution if stressful conditions were introduced? Would we always find the same thing under these really different variety of conditions?
That's a really, really, really good question, right? So the thing that sort of population geneticists worry about a lot is this thing called antagonistic pleiotropy, which is a big fancy word, which means, or it's two words, it's a compound noun, which means you can get better under one set of circumstances, but you're almost invariably going to get worse under another. And so it's exactly your question, which is like, sure, they got better under these fancy pampered conditions where, you know, they stay in the West End Hotel and we bring them breakfast in bed every morning and nothing bad ever happens. What would have happened if they'd lived in a more robust environment? And we've grown them in some other environments and they're not obviously terribly much worse. And Marco tested them for their sensitivity to various agents that damage DNA, right? Different sorts of drugs that affect different aspects of DNA metabolism. None of them are worse off than the strain they started with. Some of them are much better and some of them aren't. And I guess that's why I like the domestication experiment, because at least at the beginning stages of domestication, they were sort of out in a field and all this stresses like drought and, you know, insects biting on them and stuff that are there in the natural environment were there. And so if those mutations made them incredibly stress sensitive, they shouldn't have succeeded. Now, you know, modern crops do get it, you know, selected in greenhouses, and they do have trouble with various things. And I guess the short answer to your question is, because it's an obvious question, we should really try hard with a bunch of stresses and we should ask two things. For these guys who have evolved to live without CTF4, are they only doing better under this pampered set of conditions? And have we actually, in a sense, made them even worse off under certain sorts of stresses? And then the other experiment we should do is we should take all the mutations, but we should have the wild type version of CTF4. So now what we've done is sort of monkeyed with a wild type strain. And we asked, have we made it sensitive to stresses, more sensitive to stresses than it used to be? And that's not a hard experiment to do, and it's really worth doing. So it's a very good question. Thanks.
Yes, Brooke?
I also have a question that sort of follows up with that. I don't know if you want to go ahead with the Q&A ones or if I can pop in.
Why don't you pop in? I'll take a read.
So I work on crops. I'm a crop geneticist, and I thought a bit about this domestication, evolutionary consequences of it. And one thing that I think is particularly true of our domesticated animals and plants, and not so true in nature, is the strength and consistency of direction of selection, and the dramatic reduction in population sizes consistently across most domestication events. And so you end up with a kind of unique set of circumstances, I think. And so I don't know if I feel like they might be a hard system to make a broader argument about, although I agree that there's plenty of these really great cases where loss of function or breaking something has ended up being really adaptive or potentially novel and useful in agricultural settings.
Yes, that's a great point. And so I guess what I'll play the devil's advocate a tiny bit, right? So there's two sorts of tempos to evolution. There's a very slow change when you have big, huge populations and complicated ecosystems competing against each other, and in its mature sort of succession ecology, big population sizes. And if you get some funky loss of function mutation that allows you to invade someone else's niche, someone's already there and they're fitter than you, and they're going to just outcompete the crap out of you. So you're going to be a hopeless monster. And there may not be that much invention, right? And I guess what I'm going to claim is, because I'm sort of a wild extremist, I'm less interested in that form of evolution than the form I'm just about to describe. So the form I'm about to describe is illustrated by the Hawaiian Archipelago, right? So it lies there in the middle of the Pacific Ocean, and a new Hawaiian island appears about every million years as the plate drifts over this hot spot and lava bubbles up and you make a new island. Everything that got there came from the continental Americas across what is, anyone who's flown to Hawaii will have noticed, is a large expanse of open water, okay? And so if you're familiar with bad European low-cost air carriers like Ryanair, the way of getting to Hawaii is much worse than Ryanair on its worst day. And so as an example, most insects didn't make it on the little Noah's Ark that went to Hawaii, but some Drosophila did. So across the globe, roughly, there's 3,000 species of Drosophilid flies. 700 of them are endemic to Hawaii, and their level of morphological, ecological, developmental diversity wildly exceeds that of the rest of the population. And so the hypothesis is that if you get to a place where there are a whole bunch of unoccupied niches, there are two possible, well, I guess there's a debate. One possibility is the people who get to take over those niches are the guys who get there first, and the other is it's the guys who get there best adapted. I'm going to side with the guys who get there first. So you get some funky ass pleiotropic mutation, loss of function mutation, that allows you to invade a new niche. You're not great, but there's no competition, right? And now, and you probably have some tiny initial population size, which, you know, reduces the effect of selection, and so you can accidentally accumulate deleterious mutations. But now once you're there in your little super new niche, your population can expand and you accumulate what a geneticist would call suppressor mutations that sort of pay off most of the costs of this mutation that produce novelty, but preserves the benefits. So that's the idea that I'm pushing. I'm pushing that sort of, crazy niche expansion, the sort of stuff that's happened in Hawaii and looks so cool and funky. Because you're a plant person, so you know about the Silver Sword Alliance in Hawaii.
Well, I was going to say, I think there's some really great potential currently happening with invasive species and rapidly changing climates that I think that would be excellent comparative system.
Yeah.
But yeah, thank you. I definitely see what you're saying.
Anyway, so I like to be as sort of provocative. I mean, I wrote a review about it, and at the end I said the intention of this review is to hopefully to provoke people enough to see if they can prove me wrong.
A question written in the Q&A is from Linda Huang, who says it's a very interesting talk, and she's wondering about mutations such as the SLD5 mutations that make wild type fitness wars. If you were to do these experiments with that mutation, what happens in terms of fitness? If I'm paraphrasing, you could also read the question.
Yeah, so I guess what I would say is, in that situation, the SLD5 mutants make guys wild type a little bit worse. And so, yes, we could take those SLD5 mutants and evolve them to see what happened, right? And it would be fun to do. The problem is the smaller the fitness decrease, there are two problems. First is the slower the evolution goes. And the second is, even though yeast have been in a lab for a long time, we don't typically propagate them exactly the way we do in these experiments. So if you just take wild type yeast, and Marco did this as a control, I just don't show it when I give the talk, and you evolve them in parallel, they get over the course of 1,000 generations about 5% fitter, right? To a first approximation, everyone and their uncle has done this experiment, and there's been a lot of sequencing, and we did it too. It's a different set of mutations. But the smaller the fitness defect of the damage you've done, the more you're going to start mixing up the mutations that fix the fitness and the mutations that just make things grow faster in standard lab medium, which is why, interesting as though that experiment is, we're probably not going to do it.
This is a question from Rick Caselli. He asked, I can see novelty produced by gene inactivation if it follows duplication, in particular in plants which you cite where polyploidy is extensive. Have extra copies allowed the novelty by inactivation some copies, by inactivating some copies?
Yeah, so that's a great question. So our guy, budding yeast, actually arose from what's called a whole genome duplication. So sometime between 25 and 50 million years ago, two fungi mated, two yeasts, and they were sufficiently genetically different that their chromosomes were not going to recombine properly in meiosis. And so they were going to make dead spores. So it was going to be post-zygotic reproductive failure. And then what happened is they endo-reduplicated. So now it's a sort of pseudo-tetraploid. It has two copies from one genome and two copies from the other. And now they can go through meiosis properly. And in the intervening time, we're going to say there were roughly 5,500 genes in that original strain. For 5,000 of them, you just lost one of the two copies. For 500, Saccharomyces cerevisiae, that's the Latin name for budding yeast, has kept both. And their function has become sufficiently different, I think lots of times quite subtly different, so that you kept both of them, so that they've both been under selection. So both gene duplication and genome duplication have been potent reservoirs for sort of evolutionary novelty, and particularly for duplication and divergence of genes. And I guess it's an interesting question, going back to the question about are we studying this sort of extreme version of evolution, whether the sort of thing I'm talking about, like gene inactivation, occurs in these extreme circumstances where there is real pressure to evolve fast, and other things like slow progressive improvements in genes and the sort of polyploidy that you're describing are much more important in more sort of complex succession ecology.
Maybe one more question from Rob Stevenson, somewhat a long one there. But he asks, often we use a machine algorithm metaphor to describe the functioning of life. Your experiments indicate that if you break one cog or remove a line of code in the machine can still function. Of course, we need this kind of property for evolution to work. What new metaphor should we use to describe how these tolerant the machines of life are to defects?
I think that's a really spectacular question. So I'm incredibly interested in the similarity between human engineering and particularly software engineering and biological systems, right? So when I think about evolution, I always think about it, and I've gotten myself into trouble this way because it starts to sound like you're talking about intelligent design. I think about it like, I imagine an organism as having volition and consciousness and like, let's work out the best crafty strategy to do stuff. And what would that strategy be? And then I have to think like an evolutionary geneticist, how can that happen by just the random accumulation of mutations and selection? But computer code, like, organisms are vehicles to disperse genes in time and space, and computer code is to make things like Zoom work, right? And what I find especially interesting is they both have this incredibly restricted syntax. So computer code is ones and zeros, and biology code is A, G, C, and T, right? And then you write software that does all sorts of cool and crazy stuff. And, and I think there's an idea of modularity in biological function that's, exhibited by modularity in computer code, like, in your favorite word processing software, there's a spell checking module, and, some combinations of dudettes and dudes, sat in some windowless basement at Microsoft and primarily worked on that and had to integrate it with the rest of the machinery. And I guess what I'd say, one answer to your question is, sitting in my driveway is a 21-year-old Volvo 850. I cannot tell you how many different aspects of that car are broken, but it still runs. Actually runs pretty good, right? But the odometer stopped working. The kids and I were really excited. We were going to get to 100,000 miles. We were so psyched about this. And at 98,105, the odometer stopped working. Tape deck stopped working. The passenger side window, the electric motor is broken. The ceiling insulation is starting to sag down. There's all sorts of stuff wrong with that car, but it basically still runs because the absolute essential innards of it are relatively small. And my guess is that, one answer to your question would be really to go talk carefully to engineers, both software engineers and sort of hardware. I don't just mean computer hardware, but, like physical object engineers, and ask, how often have people figured out how to do something better because some part of some machine broke and it still worked and perhaps even had some interesting property? And I bet the answer is non-negligible. Right, so I think there is sort of an analogy between the two, and that, one thing I say to students when I talk about the cell division cycle is like, one of the things that helps when you study things is to study things that are really built for one purpose. So imagine you came from Mars and you wanted to find out how cars work. So the other car in the driveway is a 4-year-old Honda minivan. Okay. It has all sorts of stuff that I don't necessarily need. and it has safety things like I can't start the car unless my foot's on the brake, right? So if you come from Mars, you'd be really confused about what the brake did, because you both have to press it to start the car and to stop the car, right? And what I say to students is like, if I came from Mars and I wanted to work out how cars work, I go to a drag strip and I'd watch a, proper drag racer, because all you got is a big engine, some big tires, transmission, and it's just designed to go, and actually pretty much exclusively in a straight line. And so I think, I mean, a question I'm super duper interested in is what's really the minimal set of things that are required in the most pampered circumstances to keep a cell alive. And there's this spectacular, spectacular example that probably many of you guys know about. So there are bacteria that live inside the cells of aphids, which are the things that stick their sharp spike into roses. The aphids have been living, the bacteria have been living inside the aphids for 150 million years without ever going outside to take a stroll about, okay? So they're down to 600 genes. Essentially, every piece of regulatory machinery has been jettisoned, right? And the reasons it's been jettisoned is they live in insect cell cytoplasm, and the insects are sort of worried about the homeostasis of that cytoplasm, so someone else is actually taking care of the regulation for the bacteria. And so it's a really interesting question about, what's the irreducible minimum set of things that are required to keep a cell alive? I'm super fascinated by that question.
Wow. Well, I think that deserves a beer.
I'm going to come back. Is it the same link or a different link?
No, so for people who are still here joining us, I put the link in the chat.
Okay.
And so please join us. We'll take a short break, maybe go to the refrigerator, but we'll continue our conversation. Andrew will be joining us at this happy hour. So everyone, welcome. Thank you, Andrew, again for this
really
fabulous, interesting talk.
Thank you.
So we'll see.